My name is Johannes Schmid, and I will show you now how NetworkAnalyst can be used for multiple gene expression data and meta-analysis. So we start with the startup page of this web platform, and then we click multiple gene expression data, where we enter in this window. Here we can then upload different data sets that we would like to compare. So we go here to upload, and then we choose a data set of interests, like smooth muscle cells control versus interleukin-1. We submit that. Takes a little while. And now we have to go through the different steps of ID conversion, annotation, visualization, and so on. So we click subsequently on these different active buttons. First we have to go to processing. We have to specify the organism, which is human in this case, and the gene identity is official gene symbol. We click to process. It tells us here that 15,972 were matched, 1,000 yeah, we have to cut that out afterwards. Anyway, okay. So then we click to done. The next step is the annotation of the data. So we have to choose a condition. So basically we have just one condition here, treatment. The group labels are here control versus interleukin-1 alpha. We submit that. Now we can go to some kind of diagnostics, visualization of the data. So we will see the box plot showing that actually the samples are quite homogeneous. When we go to BCA plot, we will see principal component analysis for the different columns of the data set. So the next important step is normalization. So we click to normalize. This is RNA sequencing data. We perform a log2 counts normalization. We click submit. So the next step is then differential expression analysis. There we have actually only these two options because there are only two treatments here in this data set, control interleukin-1 alpha. We set the p-value and then we click on submit. So 251 genes are significantly altered, the rest is non-significantly changed. We click on done. So finally we have here a data summary that we could view. So with this we see the number of annotated genes, the number of samples, and how the normalization was done, and how many differentially expressed genes had been identified. So with that we are basically finished with this first data set, and now we have to add the other data sets. I do that now quickly without further explanation. So upload comes first. And now I go again through these different steps. Human is already predefined now. Normalization should be then exactly as before. Now we go to analyze. Here's a higher number of genes that is significantly altered. We can again look at the data summary. Now finally the third one, so we are basically comparing the response of smooth muscle cells to interleukin-1 alone, PDGF alone, and the combination of the two of them. processing. Annotation. Here actually we have to go to treatment because there was another class defined in this column. Normalization. Analysis. Here the number of significantly altered genes is much more significant. You can look at the data summary here again. And finally we have all the data sets included. So now we look what happens when we click on proceed. And here we can actually, Okay, so it tells me here integrity check failed. There are different group labels that we included here. So we have to use the annotation cell to edit the group labels to make them consistent. So we have to go here to cancel. So let's look at the annotations table. So here we have control interleukin 1, that looks okay. Here we have control PDGF, that looks okay as well. And here the third one, that looks now different. Probably that is the reason why it did not work. Okay, doesn't work, still doesn't work. I think I have to close the window now.